Hi, this is Damon, a PhD candidate in Watts Lab in UMass Chan Medical School. I'm presenting G-rich motifs within phosphorothioate-based antisense oligonucleotides drive activation of frataxin expression through indirect effects. Friedreich ataxia is an incurable disease caused by frataxin protein deficiency, which is mostly induced by G repeat expansion in intron 1 of frataxin gene. Several papers have been published using repeat targeting ASOs for activating frataxin expression. Here, we set out to develop non-repetitive ASOs to activate frataxin expression by targeting within the first intron of frataxin gene. We synthesize ASOs targeting both upstream and downstream sequences of the G repeat region and screen them in patient-derived fibroblasts. We identified two hotspots for activation, and in each case identified multiple ASOs able to increase frataxin mRNA expression by twofold in patient-derived fibroblasts. We verified by dose-responsive studies, multiple normalization controls, and two independent RNA quantification assays that the activation was not a normalization artifact. The frataxin activation by identified ASOs was consistent in multiple cell models, including a second patient fibroblast model with over 500 GA repeats and multiple wild-type cell lines. Thus, the activation was independent of GA repeat length. To understand whether this ASO-induced activation is independent on RNase H, which is key regulator of R-loop homeostasis, we silenced RNase H1 using siRNA and tested it S10 and S30 ASOs. Those ASOs can still activate frataxin expression to a similar level compared to the control, indicating this ASO-induced frataxin activation is independent of RNase H1. To understand this more comprehensively, we conducted an RNA-seq. Surprisingly, RNA-seq revealed that S10 and S30 derived ASOs could drive a unique transcriptomic profile in patient cells, which did not resemble that of wild-type cells. This made us question whether this was an on-target effect. To study this, we then deleted ASO binding sites by CRISPR in 293 T cells and established single clones. S10 and S30 derived ASOs were still able to activate frataxin expression, indicating this was mediated by indirect effects. Further RNA-seq analysis did not identify directly base-paired off-target genes shared across ASOs, although these two ASOs induce very similar transcriptomic profiles. Non-hybridization-mediated effects are often driven by protein binding associated with the negatively charged PS backbone. Thus, we tested S10 and S30 ASOs in PMO backbone, which was neutral. These PMO ASOs cannot activate frataxin expression, indicating the activation is dependent on PS backbone. Mismatch studies identified two guanosine-rich motifs, CCGG in S10 and G quadruplex in S30, that were required for frataxin activation. Here are the conclusions. I want to highlight that our study demonstrates the importance of multiple detailed controlled experiments and target validation, which can minimize the risk of advancing false positives into further development, thus improving ultimate success rates in developing oligonucleotide drugs, particularly those utilizing novel mechanisms such as gene activation. Thank you.